Hello. So how are we doing? Scared? Stressed? Oh no, I totally get you. Did you have a chance to look over the PowerPoint that I made? Yeah, was it helpful? Yeah, no problem. I'm glad it helped. I know a few of the slides are like you just need to know this, know this, know this. It's not a whole lot of like there's not a whole lot of ways I could ask you a question that's how do I say this? It wouldn't allow you to critically think about it. It's more like you just need to know the process and then you might get asked about the process, you know? So I'm glad it helped. And then I had a quick question. Do you currently go to the lectures in the morning? Good. Keep it up. Yeah, that is probably the one characteristic of high B and A students is that they always go to the morning lectures. So you're doing a good job setting yourself up for success right there. And I know when you're looking at the material, you just get like, I need to know this, I need to know this, I need to know this. It's, you know, you're bouncing back and forth a lot. And it's kind of nice to have everything in one place, look at it, study it. But yeah. But yeah, let me write down a couple of things real quick. Let's see. All right. So did you have any specific questions or anything that maybe was on the slides or anything you saw in like the PowerPoints, anything said in class, lab material, anything you had a question on? Yeah, let me pull up that PowerPoint real quick so that I can show you. Let's see. Thank you. I was about to say, is it chapter 6, 7, 8, 9, 10? Which one is it? But yes. Oh, and I also have a good little way that I remember if you're having trouble with memorizing the specific man, words are not coming easy today. Antimicrobials that target the ribosomes. If you're having troubles memorizing, if it's like this one targets the 50S, this one targets the 30S, this one targets whatever. I have a good little way to memorize that one, and I can show you that if you'd like. Let's go down to the ribosome. Okay. Let's see. Okay. So, remember that translation? Oh, sorry, I didn't look at your chat. Yes, I will. Okay, perfect. But translation is all about the ribosomes, right? So we have our bigger 50S subunit and then our 30S on the bottom. The 50S subunit is going to have our P site and our A site where the codons are going to come through, and that's our mRNA. So we have this ribosome that's kind of, it's just kind of holding this together. I kind of like see it as like two hands, like pulling along this strand and moving it along. So we have that part, right? We have the beginning set up. And then I want to go down. Okay. And then what happens is we have our start codon, right? That's always AUG. So AUG, it codes for methionine. And when that start codon is in the P site, which is just where the attachment site is, our tRNA is going to come in with the anticodon. It's going to be complementary and they're going to bond together, right? And it's going to allow for the amino acid chain to begin. So they're going to momentarily bond. And then once it moves down the side again, so once the second amino acid comes in to the A site, it's going to kind of move down. It's going to form that bond between the two amino acids, and then the extra tRNA is going to just break off, right? This peptide bond is going to form, and then it's going to shift down. And then the anticodon breaks off, and then we're just left with our amino acid sequence, and it's going to continue to grow until stop codon comes along. So it's just going to be kind of, one comes in, one comes out, one comes in, one comes out until it keeps growing. And I think one of the things that you will need to know that it's not one ribosome and one mRNA just feeding through constantly. It is like, here's one ribosome, here's another ribosome, here's another ribosome, and this mRNA is just coming through, and it's just spurting off several lines at once. So these polypeptides theoretically would all be the same. And because of this, it's called a polyribosomal or polyribosome. I want to say it helps them go through binary fission a lot quicker. Well, you probably won't need to know that one, but just do know that a strand of mRNA, it can be polyribosomal. But yeah, it's right. It's the start codon. Another one comes in, binds, they move down, and they keep chugging along. I want to say that you don't really need to know what the A site or the P site really is either. I don't think that there's any really specific questions on that one. But yeah. You also won't need to know this word, the Shine-Dalgarno sequence. You don't need to know that word.
When you're can you show me however you said you remembered the
antimicrobials?
Yes, that.
Yes, I got you. Yeah, let me pull up chapter 10 because that's where that one is. Let's see. Okay. So let me find if I'm not blind and I don't overlook it. I'm blind and I overlooked it, huh? Dang it. Oh, here. It's because it doesn't look like the other ones. Okay. So I remembered that tetracyclines, they interfere with the attachment of tRNA to mRNA. So how I remembered that one was it's gonna involve translation and tRNA, the ability of tRNA to bind. So T in tetracyclines with the tRNA specifically. For the chloramphenicol, let's see, 1, 2, 3, 4. Yeah. So for the chloramphenicol and the streptomycin, these are both part of the ribosome, right? So how I remember it's the 50S versus the 30S is that the fifth letter in chloramphenicol is an R for ribosome, and that is for the 50S portion. So the fifth letter is an R for ribosome, and the third letter in streptomycin is an R for ribosome. That's how I remembered that one in terms of this whole picture. But
Yeah, that's perfect. I needed something that was like, I don't know, not a mnemonic. I mean, it could be a mnemonic, but just something because I like can remember it better if I have like a picture in my head of something.
yeah, definitely. It's not just like, this is this, repeat it until I know it. It's, I can look at it, and even if I'm, fried from the exam, I can still figure it out. But yeah, do expect to spend at least 2 hours on that exam. That's what I heard from at least the Monday, Wednesday group I stopped by after they were done.
So is there like on the lab portion of the exam, I guess this time around, it doesn't seem like there's a lot that's going to be like physically, like compared to like the first exam where we were like streaking and all this like physical stuff. Is it more like questions based on the lab?
Yes. So essentially what's going to happen is you're going to walk in and there's going to be the stations 1 through 5, and you're going to see a folder at every single one. And it's going to be questions like, I remember one of the bigger ones, like the more heavy point wise, was it basically had a blank drawing of DNA replication and said label, like I want number 1, 4, 7, and 8. And I wound up just being like, okay, I'm gonna label the whole thing, and then I'm gonna go back and say which one exactly it wants. So there will be things like that. There might be questions about if we test chloramphenicol on this bacteria and you have a plate and it points to the chloramphenicol and it says, is the bacteria resistant or is it susceptible to the antimicrobial? You'll have to put that down. I think one of the biggest things that trips up students, and it's such like a, it concept, it's the resistant versus susceptible. You just have to read it and say it to yourself in the correct context, so that you can get an idea of what you're looking at. Because if you could always say like the bacteria is resistant to the antimicrobial, or the antimicrobial is susceptible. You could like say it in a different way, but it's easiest just to say the bacteria, does like what I did was I was like, the bacteria doesn't like this, and I would say if it doesn't like it, it's susceptible. If it is like, I can live with you, it's resistant. Just so that I could just replace the word and I don't get tripped up in my own head. But yeah, let's see. A lot of this modes of action is just memorizing. I remember it was like the day of the exam and I was like, quiz me. I just need to memorize it. Like, it's so hard.
I literally just went out to dinner with my friend because she's only in town for like a week. And I was like, the entire time I had like my Quizlet pulled up and I was like, girl, you need to quiz me on this stuff.
Yeah. Yeah, it's the repetition, it helps drive it in. I'm never a good memorizer. I feel like it always leaves. I like concepts that I can explain. All right. Let's see. Um, did she talk a whole lot about my cells, by the way? Yeah. Okay. We know what those are. Okay. Are there any other questions about like any of these? Like I want to say that the beta lactam ring and the beta lactamase are kind of pretty straightforward in terms of resistance. I want to also say that yeah, so just because penicillin has a beta lactam ring doesn't mean that's the only one, right? Cephalosporins do. So sometimes penicillins and cephalosporin can be under the same umbrella term if you just call them beta lactams. That means if they just have a beta lactam ring, right? I'm trying to think. I always think like this is like a weird one. Like it has like the name and then a letter. Like none of the other ones are like that. And it's one of the only ones that are for fungal cells. So I just remember that one like that. Um, oh, I always think about polymyxins as we're mixing up the plasma membrane. Like we're kind of shuffling it up. Um, because it does disturb that the plasma membrane. And if you think, okay, if it's more effective against a bacteria that has a outer plasma membrane, then obviously our gram negatives are going to be more susceptible, right? And because fungal cells are really different than bacteria cells, are probably not going to work as well, right? I want to say that there's not a whole lot of this on here, but probably no, that one, the pyrazinamide. I don't remember any like specific questions on that one, but yeah, I feel like these are funky. We just talked about those.
So basically, like we should just know like the overview of the antimicrobials, like how their mode of action is, like how they're for like whatever. Because some of the slides in here are like very specific about like semi-synthetic cephalosporins, and I'm like, I don't know if that's like we need to know that specific stuff for the exam.
The only thing that you need to know really about that is a semi-synthetic drug is halfway naturally occurring and halfway synthetically made in the lab.
Okay.
Like you don't need to know so much
Yes.
as to the chemical structures and stuff like that. It's
Okay.
You're fine. Know that probably gentamicin is an aminoglycoside. They're like a newer one. So yes. So these are all considered aminoglycosides, I believe. Let me fact check myself. Streptomycin, gentamicin. I don't know about chloramphenicol. That one's a weird one. But those are all specific to the ribosome, so you're fine with those. Oh, and know that like if we have a if we have two antimicrobials and one is going to target let's say one's going to destroy the cell wall, right? And the other one is going to deal damage internally. If we have that one that deals the first blow and then another one that comes in, they act synergistically. But be careful, because sometimes they treat cell wall synthesis. They're effective against the synthesis, so the growth of it. They need the growth of the cell wall to be effective against it and kill the bacteria. But another antimicrobial could have a bacteriostatic effect where it halts growth. So it wouldn't really do anything. It would halt it, and then the other antimicrobial that you're using, it could have an effect where the microbe all of a sudden gains resistance just because it's there. It's kind of like, you know this about a microbe, you know this about a microbe, you know this about a antimicrobial, you know this about an antimicrobial. Now, how do they all work together? Let me see.
Oh, do we need to remember all of the medications?
All of the
medicine?
Yes.
It's so many.
I know, it's a lot.
Thank you.
Mm-hmm. Let me see what else I can talk
So
about.
we need to remember all the medicine, but we don't need to remember the formula of the medicine, the structure.
You don't need to know the chemical structure. You just need to know the name, what it targets, and be able to critically think about how they could interact with each other. So like how I was talking about the bacteriostatic antimicrobials. So if we were to give penicillin and a tetracycline, I think that's an actual example in here. Let me see. I feel like it's an example somewhere in here, but it is like one requires growth, one inhibits growth. So how would they be able to act synergistically if that was the case?
Is that on the 68?
Say that one more time.
It's on the page 68, the slide 68.
Oh, okay. Okay, then there you go. Let me think what else I could talk about with these ones. Let me see. Know the tetracycline with the calcium ions? That might be a lecture question. The chloramphenicol last drug of choice. Know a little bit about the aplastic anemias. And how chloramphenicol basically causes that. So if you use too much chloramphenicol over too long of a period of time, then we're going to basically cause the permanent aplastic anemia. I'm trying to think.
So
Yeah.
do you think of the chapters that like chapter 7 and chapter 10 are like probably the most like significant, because like chapter 7's the DNA translation and all that, and then 10's all these antibiotics? Okay.
If I had to pick two things that are going to be the biggest things on the exam, it's DNA, all of it, and antimicrobials, all of it. Let me see. I think you need to know about the heavy metals. Something. Oh, wait, no, that was last. No, wait. Oh, one thing you will need to, another like memorization thing is all of the different ways to inhibit microbial growth. So we have our chemical versus our physical, right? And then listing out those, you'll need to know at what temperature for how long does it mean to boil and what for pasteurization, things like that. It's kind of nice because those numbers, they're a little more unique. They're not just like boil for 10 minutes at this degree. It's a little, it's like a pattern more. Let me think. Oh, yes. When something's an anti-metabolite, it's going to interfere with enzymes. So these competitive inhibitors, they're going to bind to that enzyme, and they're going to create a complex that makes the original I want to say substrate that would have bound to that enzyme no longer able to bind. And then because that's a metabolic enzyme, we no longer have that metabolic process. That means that we no longer are growing, we're no longer producing, right? Because we don't get bacteria that have these metabolic enzymes just sit there. They're supposed to do something, right? And an example would be the sulfonamides. That's the example with the folic acid and the PABA. Oh, these base analogs. So base analogs, just think about them as like a spy. They're undercover. So they kind of look the same. Okay, think about this. Think about like your normal like mafia boss in a trench coat, and then think about the three kids stacked up on top of each other in a trench coat. In the dark, they might look the same, and one might fall for it. And that's exactly what DNA does sometimes, is they fall for the three kids in the trench coat. So that's what the base analogs are. They basically just sneak in and they alter how they're read. So like you don't need to know these. These are just examples of how they can be structured really similarly and just kind of sneak in. Like this would be like this would be like two kids in a trench coat. This doesn't look like tall enough. I don't know. But yeah. That's just what base analogs are. I want to say you'll need to know these. At least just this one is for herpes simplex, as well as this one. Yeah. Oh, my boyfriend brought me pot stickers. How nice. They're not cooked correctly, but I'll give him credit for making me food.
What's your verdict on those pot stickers?
They're pretty good. They're pretty good. I can't lie. Oh, he brought me sriracha too. Hell yeah. Thank you. Shout out. All right. Yeah. To continue. So rifampin, that one, it's mostly going to ask you just about RNA transcription. So I just remember R&R. I'm trying to think of other things to remember this by, because I remember I just had to like memorize it by sheer will at a certain point because I ran out of creativity. But also, I think the only things you really need to know about this one is that it treats TB. Oh, and it's basically like How would I describe this? I want to say it's like that one kid in the group project that always works together with other people and like, you know, they always want to get the project done. They want to get it done super quickly. They outline everything. They tell you exactly what to do. All of the above, right? They interact with this specific drug interacts with a lot of different drugs very easily. Hmm. Hmm. I
Okay,
want to
personally,
say,
I think those were all the questions that I had.
oh,
So I'm just gonna go just because my cat is harassing me. So bye.
bye. Have a good
Thank
one.
you.
No problem. Well, Ling Ling, do you have any questions for me?
No,
No questions?
I think I just need to remember all the medicines on chapter 3.
I know. It's a lot.
Yeah, I just finished to review the video. I need to remember them.
Mm.
If you are busy, you can leave because I can't live here for a long time. My baby should go to sleep.
Oh, okay. Well, you're no problem. You're good to go. Thank you. Bye.
Thank you. Bye.